At CellNetix, we're fortunate to have a very large number of pathologists in our group. What that means is we're able to pool our professional resources. Although many of us have general pathology training and sign out general pathology, a lot of us also have subspecialty training. So, for instance, in my particular field, I specialize in dermatopathology. We have 4 board-certified dermatopathologists, and each one of us are capable of providing accurate and succinct diagnoses. Because we're at CellNetix, we're able to pool a lot of our resources. We're able to have a state-of-the-art laboratory. And with that, we're able to develop a lot of ancillary tests. One particular type of testing is called immunohistochemistry, or IHC for short. So IHC is a type of methodology where you're able to look at a population of cells under a microscope and determine what type of protein markers they express. And from the combination of different expression markers in these cell populations, you are then able to get a better sense of the lineage or the type of cell that you're looking at that is not readily apparent just by looking under the microscope. In addition, sometimes the IHC markers will give a specific diagnosis, and sometimes it will give me a sense of the biological potential of a particular diagnosis. Another example is our direct immunofluorescence lab, otherwise known as DIF. Now, DIF is similar to IHC, which I spoke with before. DIF is important in the differential diagnosis of blistering diseases. It helps us delineate the molecules within the basement membrane zone of the skin. And using the patterns of expression with the DIF test, we are then able to tell you whether or not this particular skin lesion is an immune-mediated type of reaction or not. And furthermore, it allows us to make a distinction of the different immunobolus disorders, such as pemphigus versus pemphigoid, versus IgA-mediated immunobolus diseases, for example. Our goal at CellNetix Dermatopathology Service is to have at least 94 to 95% of our cases to be turned around and signed out within 48 hours after receipt of the specimen. I don't believe in reports, I believe in patients. So whenever I render a diagnosis, it's really helping the clinician treat this patient. Whenever we render a report, we always think about the patient behind this report. And it's not so much the report, but it's rendering a diagnosis to help the clinicians help this patient. So we truly think that we're part of the clinical team, in a sense, when we're collaborating with the clinicians to help the patients. CellNetix cares. We care about our diagnoses. We care about all the processes we put into it, whether it's from the logistical side and making sure that your specimens are accessioned correctly and are not misidentified, to the professional aspects of rendering accurate and succinct diagnoses. We really believe in our work. We take great pride in it, and we're always troubleshooting. We're always improving, and that's what I love about CellNetix.